My name is Marit van Gils. I'm a postdoc at the University of Amsterdam, and today I'll present my data about a new broadly neutralizing antibody that I've been isolating from an elite neutralizer and that targets a novel site at the GP120-GP41 interface. More than 30 broadly neutralizing antibodies have been isolated recently, and these antibodies can neutralize variants from different subtypes, and these antibodies have called broadly neutralizing antibodies. And we've known from many studies that these antibodies can recognize different epitopes on the viral spike, such as the apex, the CD4 binding site, as well as interface antibodies and the ones targeting the glycan site, the N332, for example. And from recent challenge studies, we've seen that there is a direct correlation with the in vitro neutralizing capacity of these antibodies and their protective capacities in monkeys challenge studies. And so these data suggest that eliciting this type of broadly neutralizing antibodies will help vaccine development. And so we want to elicit these type of antibodies as an ideal to get sterilizing immunity globally. However, how to elicit these type of antibodies, we do not know yet. So from natural infection, we can learn a lot. So for many years, we have been working with the Amsterdam cohort studies and identified many subjects, both in the intravenous drug user cohort and the MSM cohort, and shown that some of these patients can really neutralize different viral strains very potently with high titers. And so these patients are very interesting to see how these antibodies are developed, and we can learn how to also elicit these type of antibodies in the vaccine. So when we look at these individuals over time and see how these antibodies are developed, we see that especially these elite neutralizers, as we call them, with very high potencies, also develop their antibody responses very rapidly. And already within the first year of infection, we see a high potency neutralizing various viral strains from different subtypes. So these ones can be really as a template for vaccine development. Therefore, we want to isolate monoclonal antibodies at 3 years post-seroconversion to really see what type of antibodies did these individuals develop, and then we can go back in time and see how these antibodies were developed, as well as how the virus evolved over time. So we have a co-evolution of antibody and virus development, which can be then used for vaccine development. The method to isolate these monoclonal antibodies have been developed in the Burton lab by myself and others. And what we have used is the BG5F SOSIP trimer. So we can really fish out the antibodies that can recognize the native-like structure. So what we did was took PBMCs from the PGT145 patient, and we looked at the memory B cells, so the CD19, CD20 positive cells, IgG positive cells, and then looked at which ones recognizing the HGV BG5 SOSIP. And we're only interested in the ones that recognize the trimer and not the ones that recognize the GP120, as we know that PGT145 only recognizes trimer, and to see if this method is valid. So what we found was antibodies that could neutralize viruses very potently with PGTM 1400 in red, showing to be a very broad and potent antibody. Compared to other broadly neutralizing antibodies, it's the most potent and broad antibody published at the time. What we see is that as the PGT145, it recognizes the apex of the trimer very nicely. So with this method, we also want to look in our patients, and we took one of the elite neutralizers of the Amsterdam cohort studies and did the same method. So we took the PBMCs, looked at the memory B cells, IgG positive cells, and then went on and looked which ones were HGV specific. And we took the BG5 positive cells and cloned these to really test and characterize these antibody responses. From these BG5 positive memory B cells, we've isolated ACS202, which is a new broadly neutralizing antibody that can recognize different viruses from different subtypes. And what we see is that this antibody can neutralize various viruses from different subtypes with a breadth of about 45% and really neutralizing viruses from all different clades in a 75 virus panel. And when we compare this breadth with other antibodies, we see that the breadth is similar to other broadly neutralizing antibodies, as well as the potency, a bit maybe less potent as others, but very similar. But then was the question, what is the target epitope of this antibody? So ACS202 was tested in ELISA against the SOSIP trimer, as well as GP120. And what we see is that the antibody can only recognize the SOCIP trimer and not the GP120. In addition, we want to know what the epitope was, so we did competition analysis with SPR. So we tested competition on SPR with the VRCO1, the CD4 binding site antibody, and also other antibodies. And what we saw is that there was no competition with the CD4 binding site antibodies, but definitely there was competition with the interface antibodies, the PGT151, 35022, and 3BC315. So this directs towards that this new antibody is targeting the interface. To further characterize the specificities and characteristics of this unique antibody, we tested as well the binding for uncleaved trimers, and what we see is that this antibody cannot bind to uncleaved trimers anymore. In addition, we also tested SOCIP produced with KIF and enzyme, and this means that there are no complex glycans anymore on this protein. And what we see is that this antibody is almost not able to bind to this protein anymore, so it really needs complex glycans for binding. In addition, we knocked out several glycans to see which ones were responsible for this effect, and we saw that the glycan at position N88 really had dramatic decrease of binding, a similar curve as with the non-complex glycan SOCIP. So this means that probably the N88 glycan is really important for binding for this antibody. To really make sure and to understand what is the epitope of this new antibody, ACS202, we tested multiple mutants in the JRCSF virus. We made 60 mutants and tested this for neutralization, 34 mutants in GP120 and 26 mutants in GP41, including several knockouts of the glycans. And what we see is that, also indicated in the ELISA, is that the N88 glycan was very important for neutralization, as well as residues surrounding the N88 glycan. In addition, we see that mutation at 229, the position is not a glycan, is also very important for neutralization, and this residue is very close to the N88 glycan. Very surprisingly, we also saw a lot of effect with mutations in the fusin peptides. This is never seen before for other neutralizing antibodies, and so it's very interesting to see what the effects are. And as a control, we took PGT121, and there was no effect on these mutations. So it's really specific for this antibody. And when we map all these different mutations that had an effect on the neutralization capacity, we saw that these all clustered together at the interface of the trimer. And really they're surrounding the N88 glycan, as well as the fusin peptide that's quite accessible in the trimer. And in parallel, we also produced SOCIP trimers from viruses isolated early in infection of the same patient. So what we did was have a SOCIP from early viruses called AMCO11 SOCIP, and this was characterized in cryo-EM by the Ward lab at Scripps. And we see that this SOCIP is a well-formed native-like structure and very comparable to BG55. So when we have the overlay with BG55, we see that the structure is very similar, and therefore means that this SOCIP is a very nicely forming trimer and can be used for several methods as well as immunizations. So what we did was we made complexes of this trimer with the ACS202 autologous trimer, antibody, sorry. And what we saw is that the autologous combination had really nice binding kinetics, especially the off rate of the antibody with the ACS202 autologous SOCIP, the AMCO11 SOCIP, was much lower. So when we made the complexes and looked at those with negative stain EM, we see that there is a nice binding of the ACS202 antibodies to the SOCIP in a 3-to-1 ratio. And when we made a modeling of the class averages of these images, we see that the ACS202 has a really distinct, unique binding to the envelope trimer compared to other interface antibodies like the PGT151 antibody, 35/22, and as well as 3BC and 8ANC195. So when we look at the footprint of the new antibody in red, we see that there is some overlap with PGT151, 35/22, and 3BC315. However, it's very distinct from these antibodies, therefore saying it's a new epitope. So in conclusion, we can say that the ACS202 antibody is a novel GP120 GP41 interface antibody with a breadth of about 45%, neutralizing viruses of different subtypes. Very interesting is to see that it competes with the known interface antibodies, but with a unique footprint, dependent on cleavage, complex glycans, especially the NADH glycan, and trimerization. So ACS202 highlights that the fusion peptide is a new site of vulnerability on the envelope glycoprotein. And therefore, also the question arises, do we want to have the fusion peptide accessible on immunogens or not? And therefore, it's also very interesting to look at this elite neutralizer and see if we can really study the co-evolution of virus and antibody development. And therefore, these new SOSIPs, AMCO11, can be used as well as lineages, immunogens. And therefore, we still want to investigate this further. This whole study has been done as a close collaboration between the Amsterdam Medical Center as well as the Scripps Research Institute in San Diego, under the supervision of Rogier Sanders, Dennis Burton, and Andrew Ward. All right, thank you.